I would like to really thank all the organizers for this GlowMind webinar, for this opportunity to speak to you, and to say that I'm deeply humbled and honored to be included with such a group of extraordinary scientists. So today I'm going to be talking to you a little bit about sequencing outside of the lab. And of course, I don't do all this work without the help of lots of collaborators. Some of them are shown here, as well as some of the funding agencies that have supported the work I'm going to show you today. So let's see what we can do. There we go. We've heard a lot about molecular tools to kind of profile the microbiomes, and the bulk of all the work that's been done over the last 30 years, 35 plus years, has been looking at the 16S ribosomal RNA gene, as we've been hearing about during the webinar. But today I'm going to talk to you about a different kind of methodology, something somewhat different here. And that is using the Oxford nanopore MinION and ribosomal operon profiling to characterize the microbiome. And the reasons that I like using this system are shown here. Essentially, it's fast. It can be relatively cheap, especially if you consider the costs of buying the machine and service contracts, things like this. It directly provides the sequence information. You don't sequence by synthesis. You actually read a signal off of the DNA molecules themselves. It doesn't require extensive training in order to generate the sequence or to analyze some of the sequence. I'm a simple person. I like doing things as simply as possible. We've shown that it can be quantitative. If your laboratory practices are careful, you can get some quantitative results. And the most important thing here seems to be that it's transportable. So the MinION itself is a small handheld DNA sequencer. It's about 90 grams. I'd like to thank Dr. Egley for introducing the MinION during the webinar. We're sharing some similar figures. So the way that nanopore sequencing works is there's a series of 2,000 protein channels embedded in a semiconducting membrane. And the chemistry is created so that the DNA molecules can flow through these channels. And the electrical signal, the current from each of these pores, can be independently collected and monitored. And the important thing here is if there's a difference in the current that's associated with the various base pairs, you can use this electrical current to deconvolute out what the sequence is for your DNA molecule. And there's a variety of software packages that come with the MinION that allows you to do the base calling. And it's really helpful having some kind of DNA manipulation package. I like to use Genius. And so the MinION has only been on the market for about 6 years. And very early on, 2014, 2015, the sequence accuracy was really quite poor, only about 60%. But just within the last 5 or 6 years, it's improved remarkably as the protein chemistry has changed, as the base calling algorithms have changed. And so here we can see that at the end of using the albacore base caller, you're talking about raw read accuracy of about 90%. Over the last 18 months, a new base caller has been available called Guppy. And at the last community meeting for Oxford, it was reported to have a 98% accuracy for the raw reads. Now, we're interested not in trying to take advantage of this long read capability to look at entire ribosomal operons. We're using this as a test case. You can use other operons as well. But the reason that we like using something like this is that the long read capability essentially gives you 10 times the amount of sequence. But really more importantly, it captures this internal transcribed spacer region, the ITS region, in between the small subunit and the large subunit. And all of this contains strain-specific information. And Finally, you can actually easily characterize, these reads just using programs that we're all pretty familiar with, like BLAST. And it turns out Oxford actually has a graphic user interface system called Epitome that uses a classifier called WIMP, what's in my pot. Now, I'm not the first to be looking at ribosomal operons. There are a couple of other labs that have been doing this. The first person to publish was Alfonso Benitez Paez's lab, and we've uh, been right behind them. And the equipment that you would need in order to use this type of platform to sequence in the field is actually shown in this figure. It fits in a suitcase. What you need is essentially a MinION, which is hooked up to either a laptop or a computer and normally would be connected to the internet in order to function. But with another purchase, you can get a unit called the Minute, which is a separate computer that allows for data collection and base calling. And you're also going to need a variety of other bits of equipment to sequence in the field, like a bento lab here for extractions and PCR. We're trying to design the ability to analyze these reads on a portable system like a Mac Mini, hooked up to an iPad as a keyboard and monitor. And you're going to need a portable hard drive in the terabyte range to capture the large amount of data that the system can generate. Now, of course, once again, we're not the first to be doing this. This has all actually been pioneered by people like Joshua Quick and Nick Lohman, who did Ebola epidemiology. But this portable system has been used to generate sequence in the high Canadian Arctic, in sub-Saharan Africa, even been used on the International Space Station. So there's a number of questions that kind of arise if you're interested in doing operon profiling, and that is, can we really easily achieve species or strain level resolution with the MinION? Can you find some kind of screening method that will show this strain level resolution without the need of connecting to the internet, using servers and other things? And then finally, can you combine some of these profiling methods with other approaches to link the phylogeny and function to kind of improve sample collection and deposition in a microbial vault, where you have a better understanding of what the organisms are actually consuming, producing in order to culture them and preserve them. So before I talk about this just a little bit, I want to at least explain the importance of strain level resolution. And this comes from a lot of pangenome studies. Here's just one where 3,000 different pneumococcal strains were isolated from donors around the world. And when they sequenced all these genomes, they discovered 37,000 different genes in this particular strain. But if you look at the core genome, which is shown here, it represents only about 16% of all the genes that could be detected. So if you are not resolving strains, you are not resolving all the potential genetic variation. And you could be losing the mechanistic understanding of the biological interactions, let's say between hosts and various members of the microbiome. So we were following in Alfonso Benitez Paez's footsteps, and we generated a ribosomal operon database with collaborators at the Department of Defense, looking at complete and partial genomes to pull out these operons. And we right now have a database of about 300,000 operon reads. The bulk of this information is at the strain level. There are about 10,000 different species represented in the complete genome, 140,000 different strains. And so what we wanted to do was to first take a ribosomal operon profile that we had and just try and figure out the limits. And here we've blasted these, and you can see that the alignment length is about 100 or 1,000 base pairs, and the identity ranges from 70 to 95%. So in order to figure out whether you could reproducibly get strain level resolution, we took two entries from this database, mutated them in silico for these type of parameters, and then tested whether we would get the proper hit. And then ultimately, we would look at other representatives of the database. So here's just a quick example of what I initially picked, Vibrio coriolyticus strain 58 and strain Snooty. These mutations were done in triplicate. They were all characterized, and we knew what the percent identity was. And then we could go back and blast this against the Operon database. And in the green box on the left, you can see the identity of the reads. On the red oval, you can see the best blast hit that shows up. And so here, when we're at 82% or higher identity, we still can resolve to the strain level for this particular Vibrio. These are just some of the various blast settings. We heard earlier about how important understanding what those settings are when you're doing your workflow. This is the one that we settled on because it turns out that you could roughly analyze one read in about 3 seconds on a Mac Mini in the field. Now, if we optimize this just a little bit more and then test everything else, what you can see is that from 84% identity on up, we can resolve strain level differences. And that's whether the database has as few as 10 or 11 operons all the way up to 426. So let's take a look at just some actual samples that we would be considering characterizing. So we had published work on looking at the operons within the human respiratory system. And we reanalyzed this with the Operon database. And when you do this, over 90% of the hits generally give you back a blast result. But if you limit this to the strain level resolution greater than 100% or 1,000 base pair alignment and the 84% identity, we still don't lose very many. And the bulk of the reads that we have, we can resolve to the strain level. However, if we look at other types of environments, this is from the marine sponge microbiome off of the Great Barrier Reef, we find that level of strain resolution drops pretty precipitously. And that is that only about a third of the reads are actually resolvable to the strain level. And we think that that's really a reflection of the database itself. The bulk of the database is biomedical, and we're thinking that if there are more and more environmental samples deposited, this number would go up. But what I want to do is just show you one quick example that just pulled together within the last 24 to 48 hours. This is looking at the gut microbiome of knockout mice compared to wild type. And so this is an RGS14 knockout. That's a G protein signaling pathway. And you can see here in the pink are the wild type mice, and in the yellowish are the knockout mice. And this is looking at 3 different strains of Akkermansia muciniphila. And it's very clear that the wild type have one strain, that the knockouts have a different strain. And all of this would be lost if you group at too high a phylogenetic level. Now, the nice thing about the Operon database, since it's based on complete genomes, it's possible to now pull out that genetic information to compare these genomes. And this is just one spot. It turns out the wild type genome is complete, but the knockout genomes for the Akkermansia strains are not. But this is about 400,000 base pairs. And you can see this gap that exists in the genomes of the knockout strains that's not there in the wild type strains. And we hope that this will allow us to focus in on ways of isolating and understanding the biology that's contributing to strain selection. So let me just conclude really quickly and say that clearly the MinION, these long reads in the database, can achieve strain level and species level resolution. We can try and link that to complete genomes. The low cost of this platform, it's only $1,000. It's highly portable. And the standalone nature should allow for microbiome studies in remote sites with very little infrastructure, just power needs. And as more and more of these genomes get deposited, we should be able to improve our resolution of environmental samples. So I got a minute left. And I'd like to just make a plug for going in future directions as well, coupling all of our understanding of microbial diversity with activity measures, because that's going to help us culture these organisms. And the nice thing about the MinION is you can actually sequence RNA directly and DNA as well. But the last thing I want to show you is just coupling this operon profiling with activity measures, such as stable isotope probing. So when you do stable isotope probing, you add a 13C substrate that gets incorporated into the labels, into a subset of the population, and they make 13C DNA. You can physically separate that, you can characterize it. We've done that in soils. And so here's an example of cranberry soils that have low and high productivity, and we can determine the amino acid active bacteria and compare them with active fungi. So I'm done. Thank you so much for your attention, and I look forward to any questions.